So welcome to the lesson notes, guys. Welcome to the lesson notes. So I'll take the learners, and maybe the teachers will also look at what I have done in this area. So using a video, I've carefully looked at the points which are both significant as far as students are concerned, examination-wise and day-to-day life way, so that now, in the wrong hand, this topic is going to benefit them in their day-to-day life. So, welcome to the lesson notes on the topic cell physiology. Welcome, guys, to the topic cell physiology. Welcome, guys. So, the lesson notes, cell physiology for junior secondary students. We have introduction to cell physiology, where I start with the definition. Cell physiology is the study of the functions and the processes that occur within living organisms to maintain life. The study of the functions and processes that occur within the living cells in living organisms to maintain life. Guys, and left the word cells because cell physiology takes place inside the cell. The physiological processes, we begin with diffusion and osmosis. Diffusion is defined as the movement of molecules from an area of higher concentration to an area of lower concentration until they are evenly distributed. Example: perfume spreading in a room, oxygen entering cells, and carbon dioxide leaving cells. So guys, it's very common, especially during Sunday, where you find yourself looking to answer ready because he has applied perfume of whatever nature. Some are very nice smelling, others, you know what is you have found. But that process, that perfume coming from that ready to you, that process is what we are calling diffusion. Because the molecules of the perfume are concentrated in the ready. And now they move from where they are concentrated to where you are, you who has not applied that perfume, and then you can smell them. Osmosis. Osmosis is the diffusion of water molecules through a selectively permeable membrane. Aerating selective permeable membrane from an area of low solute concentration to an area of high solute concentration, or in short, from an area of dilute solution to an area of concentrated solution. Examples, we have water absorption by plant roots. All right. Remember, water is having low solute concentration on the ground, and then the water inside the plant is having high solute concentration, and so the water will be forced to move up the roots. movement of water into and out of cells to maintain balance, even in human body. The key points: both diffusion and osmosis are passive transport processes. Why? They do not require energy. Excuse me. The molecules move naturally. You don't require energy to move them. They are crucial for maintaining cellular homeostasis. Okay. Excuse me, guys. All right. Okay. So we come to active transport. Active transport is defined as the movement of molecules across a cell membrane from a region of lower concentration to a region of from a region of lower concentration to a region of higher concentration using energy. Remember, the other two are passive. They don't use energy. But this one is active because it is using energy from ATP. ATP means adenosine triphosphate, a very rich energy food in the bodies of living organisms. Examples are the sodium-potassium pump, where sodium ions are moved out of the cell and the potassium ions are moved into the cell. And the other one is uptake of glucose in intestines. Active transport requires energy because it moves substances against a concentration gradient. And it is essential for maintaining concentration of ions and other substances necessary for cellular functions. What do we mean by against a concentration gradient? We mean the molecules being moved or the ions being moved are being moved to where we have more ions. All right? So it's like you are pushing something where you have too much and therefore you need to use energy. All right? It's like climbing a hill. All right? You are climbing a hill, you see you pant. But if you are going down the hill, you don't even feel it. You don't use a lot of energy like when you are climbing the hill. So that movement of ions from where they are less to where they are more is what we are calling active transport, because you require energy to move those ions. All right? Glucose in the intestines, okay, requires energy. All right? Okay. Enzyme activity. Definition. Enzymes are biological catalysts that speed up chemical reactions in cells without being consumed in the process. How enzymes work. There are 1, 2, 3, 4 things you should know for you to understand how enzymes work. You have to know what is a substrate, you have to know what is the active site, and you have to know the enzyme substrate complex and then the product. So substance is the molecule upon which an enzyme acts. The active site is the specific region on the enzyme where the substrate binds the enzyme. And then the enzyme substrate complex is the temporary complex formed when an enzyme binds to its substrate. So when the enzyme fixes itself on what is being worked on, that one is enzyme substrate complex. And you remember enzymes work like keys and locks. So the moment the enzyme fits and binds on the substrate, it can break that substrate in what is required. So the product is the molecules produced from the substrate by the action of the enzyme. Factors affecting enzyme activity, we have temperature. Here, enzyme activity increases with temperature up to an optimum point, beyond which the activity ceases. So there is temperature for every activity. If you exceed a certain optimum point, then the enzyme will not work. It will be destroyed. So enzymes work within an optimum temperature. pH. Each enzyme has an optimum pH at which it is most active. All right? Like the one in the stomach known as pepsin, that one works best at pH 2. And of course, you know the temperature of the human body is around that is 6.9 or that is 6.7 degrees centigrade. Substrate concentration. Increases substrate concentration increases enzyme activity up to a saturation point. So as you put more substrate, if you put excess, it does not mean that the enzyme will work. In fact, it will be affected because you are not supposed to saturate the substrate. Enzymes work best at a certain level or quantity of the substrate. Examples of enzymes. Amylase. This breaks down starch into sugars in the mouth. Protease breaks down proteins into amino acids in the stomach and small intestine. Lipase breaks down fats into fatty acids and glycerol in the small intestine. Key points: enzymes are specific to their substrates. They are crucial for metabolic processes and maintaining life. Key concepts: cell physiology, understanding the process that keeps cells alive. Diffusion and osmosis, passive transport process essential for maintaining cellular balance. Active transport requires energy to move substances against their concentration gradient. Enzyme activity, enzymes speed up chemical reactions with activity influenced by temperature, pH, and substrate concentration. Visual aids illustrate diffusion and osmosis with the diagrams showing movement of molecules. Show the sodium-potassium pump for active transport. Provide a diagram of enzyme action, including substrate binding and product release. Activity, diffusion experiment. Use food coloring in water to demonstrate diffusion. Osmosis, use potato slices in salt water and fresh water to demonstrate osmosis. Enzyme activity experiment, use catalase in potatoes or liver to break down hydrogen peroxide and observe reaction. Worksheet, include questions on definitions, examples, and explanations of diffusion, osmosis, and active transport, and enzyme activity. Provide diagrams for labeling and short answer questions to reinforce running. Conclusion. Review definition and key points of cell physiology, diffusion, osmosis, active transport, and enzyme activity. Questions. Encourage students to ask questions for clarification and better understanding. Homework. Assign a short paragraph on the importance of enzymes in daily life with examples. These lessons notes provide a detailed overview of the cell physiology for junior secondary students, focusing on essential processes such as diffusion, active transport, and enzyme activity. Reference books. The reference books are there. So I'll just show you the reference books. All right. Kenya reference book. Secondary Biology Form 1 students book. You have to read chapter 4, the cell, and then you read section 4.1 of that book. Introduction to cell structure and function, overview of the cell component. Then you read chapter 5, diffusion and osmosis, definition, importance, detailed explanation of osmosis and practical activities. 5.2 active transport and the significance of active transport, examples of active transport in cells, comparison between the passive transport, diffusion and osmosis, and active transport. Cell physiology, enzyme activity, introduction to enzymes as catalysts, mechanism of enzyme action, factors affecting enzyme activity. That is section 5.3. International book is Biology for Cambridge Secondary 1, Authors, Pound, Range, Richard Fosley, Anne Fallac, Publisher, Oxford University Press. Relevant chapters, as you can see them. Chapter 3, 3.1, (3.2) osmosis, 3.3 active transport, 4 enzymes, 4.3 application of enzymes in industry and medical standards and examples. End of the lesson notes on cell physiology. Subscribe to this channel. Watch the video to the end, guys, and you see the benefits you are going to get. With those few remarks, guys, may I say goodbye, and I also thank those who have subscribed to my channel, Smart_Traveler Memos, for more amazing videos like this in biology. Goodbye, guys. See you next time.